So title is we are working on Alzheimer model, particularly in the title of the work is reducing endogenous kinesin in one motor domain, ameliorate tau hyperphosphorylation aggregations and memory impairment. You can say the animal of tau. So, okay. By the concept by our boss was presenting previously, there are kinesin is a motor protein. So it is mainly working on the cargo transport. There are n number of kinesins proteins in MAMA still identified. There are 45 different kinesins were identified. All these kinesins are grouped into 14 groups, and each was characterized as a subtype, like group 1 to 14. Like that, 45 kinesins were identified, and each kinesins are mostly they are doing the transport mechanism, which means they carry the cargo inside the cells from one end to another end. We call it as plus end to minus end or minus end to plus end, like that. So it particularly in the neuronal cells, so the plus end to minus end we call it the anterograde, and the minus end to plus end we call it the retrograde transport. So this is the mostly the hypothesis. This based on this hypothesis we are working here. Like that, the kinesins are just like walking on the microtubule. They just carry the cargo. They have the light chains and the heavy chains, and the light chains carry the cargo and the walking to above the microtubule and to reach any one of the end. So in our case, we focusing on kinesin type 1. It is having Q5A, B, C, like that. Some isoforms was there. So in that we are majorly focused on KIF5B, like so different kinesins are there in our lab. We are focusing on different kinesins, and now we are working on kinesin B5B. So in general, the kinesins carry the cargo, and we're going to reach on one end. That's no problem. When there is no cargo was there, it simply folded back and touched down. While during the transport time, it utilizes the ATP. When there is no cargo, it simply stop and it's folded back. The high accumulation of the kinesin, it disturbs that environment, particularly the ATPase hydrolysis or ATPase activity also utilized by some other proteins. For example, in tau, in that region, tau protein also is there. Ultimately, when the tau getting the phosphorylations, it's lead to hyperphosphorylations. In that aspect, we are working. Then microtubule got disturbed, tau hyperphosphorylation increased, and it leads to the tangle formation, that is neurofibrillary tangle formation. So usually in Alzheimer's disease, we put a split into two parts. One is A beta mediated, another one is tau mediated. The final conclusion is it forms the fibrillary tangle, neurofibrillary tangle in the brain region. So the nerve cells got disturbed. Ultimately, treated collectively called the Alzheimer and Parkinson, whatever it is. This aspect we are working. So what we do, come to the picture, figure 1. So here, what we do, the kinesins, we classify the entire structure of the kinesin, like head region, motor region, tail regions, like that. So in this protein, we are specifically we are working on which part of the protein can interact with the tau protein or which is responsible for the disturbance. So what we do, we take out different part, like head domain, motor domain, like different part, and we perform the pull down assay with paired helical filament. That means that tau forms. So it shows that 1 to 413, that is the motor domain. So the motor domain represents that the motor domain has specific interactions with the tau. So on that basis, we carried out the research. So we carried out the research in two parts. One is in cell line part, another one is in animal model part. Animal model. In cell line, we used to do different types of cells. One is SHSY5Y. That is the neuronal cells. Then another one is haploid cells, or the hap1 cells we use. In SHSY5 cells, we simply silence the KIF5B. When the KIF5B has some interaction with the tau, if it is true, when we silence the KIF5B, the tau activity also has to give some response. So here it is that in that Western blot it is showing that when we inhibit the KIB, PHF1, paired helical filament, this is a form of tau, that is also got reduced. So it is strengthen our data like that. Okay, while focusing on the tau, the kinesin also have some interaction with that tau. On that sense, we again go with the HAP cells. We simply using the CRISPR mechanism, knock out the entire KIB, then we overexpress the tau and the mutant form of tau also. What would happen? Wherever we knock out the tau, knock out the KIB, the tau activity also got lost. But usually these kinesins doing the transport mechanism, cargo carrier, uh, it is doing some basic fundamental role. The transport is very, very important for the neuronal activity and everything. From the embryonic stage, it is very important. So what would happen? Complete knockout of motor protein, it makes some lethal. But when coming to the heterozygous or partial knockout, it has some response. So in that aspect, we are working. So the same side, so here you can see in the Western blot, what we do, we use the cyclohexamide, that is a protein synthesis inhibitor. So in the same thing, we transfer the tau and kinesin knockout. We add the cyclohexamide and stop the protein synthesis also. Then what would happen? And similarly, but tau got decreased in the knockout of kinesin. So it is strengthen our data when kinesin also got absent, the tau level also got reduced. So we overexpress the motor domain. So here in this data figure one, we concluded that the motor domain is showing some interaction with the tau. So we take and clone the motor domain in a cloning vector and followed by the expression vector, and finally we purify the kinesin B motor domain. Specifically, there are the entire kinesin protein have three domain: neck domain, tail domain, motor domain, head domain. Head and motor in between neck also was there. So we need to confirm that which region is have specific interaction with the tau protein. So in the same thing, animal model, we generate the heterozygous partial knockout of KIB. Then we go for some what is called as it motor activity. Like when the response, we give the response to the response of knockout KIB, how the character changes was happened. Example here, so these are some initial parameters. We check it out whether after knockout, partial knockout of the KIB, the mice was showing that open field test, and everything was fine. When coming to the locomotor activity, anxiety, like a fear condition and everything, and compared to the wild type, KIB knocked down, that's a little bit compete with that wild type, which means that this P301S tau, that is the tau mice model, that is a disease model. In addition to that, we knock out the partial knockout of KIB. It is showing that some improvement little bit when compared to the P301S tau model. Then look at this, the black color is the wild type, then this is the P301 plus KIB knockout model. So from the behavior activity, so we check that whether how the protein activity and everything, like soluble and insoluble tau. So we have to check that after knock the KIB, how the protein expression got vary. So it shows that KIB knockout in tau model shows that completely comparatively the reduction of all the tau epitopes like 80, 80, and everything got reduced, particularly in insoluble tau. Insoluble tau is a significant result. So it is showing that complete reductions when compared with the wild type and tau model. So it is showing that when you reduce the KIF-IB activity. The tau also got response, which means that you can control the hyperphosphorylation of tau. Similarly, the cells moving towards the autophagy. So this is the area. This is the new interesting, like how this autophagy also involving this mechanism. But this area we need to address further. So this is my presentations. Oral presentations. How the one motor protein have interact with the tau protein. So how it give the adverse effect on tau hyperphosphorylations or phosphorylations. So how we focusing on the motor protein, with the motor protein, how we can target the Alzheimer's disease. So this area we are working. So from that I'm doing this presentations. So now completed my presentations. Any questions?